So thank you for the opportunity to be the last speaker in the first forum of this year. And I'd like to start with what it means to us to unravel the code of adaptive behavior. For us, it means trying to understand how this vast network comprised of many trillions of neurons communicating with each other by short electrical pulses form those intricate, complex, widespread patterns of flickering that you see here that allow to control behavior in a way that makes the animal more prone to survive, not only in some predefined manner, but in a way that adapts to the changes in the animal's internal state and also in changes in the environment. Now, for decades, it was hard to study this question. And the reason is that we were limited technologically. We could only measure the activity of a handful of cells in certain brain areas under constrained conditions and for short timescales. So imagine you're trying to uncover the meaning of this beautiful, complex musical piece that is played by the biggest orchestra you can think of, but you can only listen to the notes played by a few players, few musicians, or follow their hand position. But over the last 2 decades, this situation changed dramatically. So now, my experimental collaborators are able to routinely record the activity of large population of neurons, hundreds to 10,000, simultaneously, at the single neuron resolution, so seeing the flickering of each one of those neurons for a long duration of time, so hours, days, long weeks now, in various parts of the brain, including deep brain areas, and to do so while the animal is freely moving and behaving in a naturalistic setting. So with this technical surge, though, there is a need for a concomitant shift in the analytical and conceptual methods we use in order to analyze these high-throughput databases and translate them into understanding and knowledge about how the system works to integrate sensory information from the environment, internal information, cognitive processes, action selection processes in order to drive behavior. So this is what we try to do, and I will take you through a maybe the simplest setting. So even in this simple setting you see here on the left, where 2 mice are left in a neutralized square environment, free to interact one with each other. Speed is 4 times speed up, so don't worry, they're not on something. Even in this somewhat lab-tailored simplistic situation, the situation is already complex. So the information from all the sensory modalities has to be integrated with the internal state across a widely distributed brain network in order to drive not only the momentary muscle activations that have to do with the momentary movements, but also those slow shifts you can appreciate between tending to the environment, not caring about the animal, then deciding to interact with the other animal in a way that might turn not very amicable at a certain stage. These evolve on a very slow time scale, very different from the millisecond or the tens of milliseconds of muscle activation. Now, as the cable you can see here in the frame foretells you, the mouse marked in red is equipped with a miniature microscope that, together with genetic expression of calcium indicators, allows us to do what was genuinely almost unthinkable 20 years ago, and that is to follow the activity, to monitor the activity of here about 100 cells in the deep structure of the amygdala, that's deep in the mouse brain, while the animal is behaving on the left-hand side. So now, if we want to connect between the behavior on the left and the neural activity of this subnetwork, 100, not many trillions, but 100 neurons, and find what's the functional meaning of that flickering that you see, we would usually use the common method of going from the left to the right. That is coming with some a priori assumption as to what is interesting in the behavior on the left. Let's say the beginning of a social interaction, looking at the neural activation pattern at that time, and looking for enough repetitions in order to verify that that neural activation is reliably connected to what we saw in behavior. What we are suggesting is that these new exciting databases offer us another entry point, basically reversing the direction of inquiry. That is, starting with the neural activity on the right, finding interesting patterns there, and structures, both as to how the different neurons are activated, but and also as to how it develops in time. And then project the structures we found in neural activity back onto behavior to form new hypotheses as to what is this function of this subnetwork we're looking at. And basically, it's as if we're saying, we don't have very good ideas. Let the brain lead us in this journey and tell us where to look and what to look at. We're not basing it on any external markers, not on any a priori assumptions as to what the system is doing, when it's doing it, or even at what time scale. So how do we do it? We start with that movie you saw on the right, so it's not as nice as the GIF animation we started with, but you can appreciate those flickering circles here that correspond to the activity of the cells in this subnetwork. And then after a bit of pre-processing, we can represent the activity of all these neurons during here, it's a 25-minute session, in this kind of heat map. So every line in this heat map represents the activity of the neuron across the entire session. Warm colors, high activation, cold colors, inactivation, relative inactivation of the cell. Neurons are stacked one above the other. So we have here a bit over 100 neurons. And time along the session runs on the x-axis from left to right. And what you can appreciate here first is that, yeah, biology is noisy. We don't see anything apparent here at first sight, but if you stare at it a bit longer and remind yourselves that if you don't do it quickly enough, we won't go to lunch, you would probably be able to tell me that you see some recurring patterns inside this. And I want to guide you a bit. Instead of looking at each line and trying to analyze the responses of one neuron and connect it to something, we instead want to look at the responses of the entire site network at a specific point of time. So basically, we want to look at slices of this heat map, colorful slices that capture the pattern of all neurons in the subset. And once we look at that, we can clearly get a sense that there are some patterns that recur along the session in a noisy matter, biology still. And so we detect all the times a certain pattern, as marked here with the red dots, and then we can detect all the times another pattern. Remember, it's the pattern of all the neurons in this subselection in the network at a certain time. So if you want a single note played by the entire instrument we were discussing before. So now if we substitute those warm and cold colors by the pattern that we detected at each time point, so now time along the session is still on the x-axis. Each dot like that is the pattern that we detected at a certain time point, and which pattern it is depicted both on its position on the y-axis, which is as random as it gets, and the color. And what you can see here is that instead of the high-dimensional representation, we got here a simple time series of discrete patterns that we can now look and analyze. You will do it quickly with me with your eyes. What it might resemble, given the metaphor I've used before, is a musical score. It seems like you could play the music that this subnetwork is playing for these 25 minutes. And the point being that now, instead of looking at each time point at the pattern that is activated, trying to link it to behavior, we can also look for higher level structures, slower structures, regularities in the data, and try to refer them back to behavior. So what you can see here is that the system transitions not between all the patterns that are available, but between a subset of them. creating to a certain extent long period of time where this orchestra we've been talking about only plays certain tones. And then, at a certain point, this is the laser? Okay, no laser. At a certain point, I will ask you, when do you think that changes? At approximately what time? Hmm? Yeah, somewhere along 300 seconds or 5 minutes, half of this talk, the situation changes. And the same subnetworks transitions between a different set of patterns. And then again, transitions back. So if we were to treat it as a musical piece, we see here certain movements, if you want. Long periods of time where there is some regularity that has to do with the statistics of the transitions possible. So this is what you very nicely pointed to us. After the first 3 minutes, there is another motif that comes and transitions between a mostly different set of patterns. And then maybe you can appreciate that there is a third kind of motif that comes up later in the session where the system for long minutes transitions between a third set of mutually exclusive, mostly mutually exclusive patterns. Now, these are very long time scales for neuronal responses. These are long minutes. This is not milliseconds or hundreds of milliseconds, and there is a regularity. So what we see is that we can detect easily that and tear apart the complex dynamics into simpler dynamics that the system just switches back and forth between them, if you want, themes of this musical piece. So not just once, but back and forth, it alternates between 3 motifs, and we can take this kind of structure and project it back on behavior and ask what it means. Now, we don't have the time to go into the behavior. It's complex, so you will have to trust me on that. What we do is we just here color the patterns differently as per the movement, the theme that they are part of. And when we look back at the behavior, we see that this blue theme occurs when the animal is doing unstructured behavior. This is a mouse alone. Now there is no other mouse. It's grooming. It's exploring the environment. Looks for surprise around. Nothing structured. And indeed, also the neuronal activity during this blue theme is unstructured. The patterns transition between themselves in a way that is pretty much random with no direction. On the other hand, the 2 other themes we find in this short session are different. If we zoom in on them, we see that the entire epoch, the entire theme is constructed of repetitions, noisy repetitions, biology again, noisy repetitions of certain sequences, small melodies that are repeated again and again and again, very structured. And if we look at the behavior of the animal during those times, we see that it performs sequences of behavior that are aimed to get a goal, sequences it has learned in previous sessions in order to attain a certain goal that is desirable. In this case, sucrose and condensed milk. Well, back to the lunch motive in my talk. And as we see and verify for ourselves that it is not connected to the movements, to the actual motor plan the animal is doing, we think we see here evidence of a mechanism that is aimed to code and maintain the animal's specific motivation to do something in order to gain a reward. And I want to remind you something. The entire talk all the minutes we have spent now had to do with the time scales of between hundreds of milliseconds, seconds, to minutes, 25 minutes of a session. But I told you in the beginning that I have collaborators that are recording the data for days, weeks, and following the same cells. What I want to point out is what we can do now that we find this order and structure for a trained animal that has adapted, we can go back and ask how this learning occurred. When did these musical motifs, those nice sequences appear in the music of this subnetwork? And we can ask questions regarding learning a task, generalizing from one task to a different task, multitasking, switching between tasks, and adapting a task to some difference that happened in the environment. And this window we hope to gain from these methods as to the adaptive code of the healthy brain also has implications when we want to understand what happens when adaptive behavior becomes maladaptive. We know from our lives that many times we do things to achieve goal. And in time and circumstances, this can become habits, disconnected from the goal they're supposed to achieve. From the outside, it's sometimes very hard to tell the difference. On us, as in mice, sometimes those habits, which are actually not such a bad idea sometimes, turn into compulsive behavior and addiction, which are already harmful for the animal and for us. During the progression from adaptation to maladaptation, many times it's impossible to know from the outside behavior at which stage the animal is. But if we're listening to the music of the brain, we can see when certain motives disappear or change dramatically. And projecting it very hopefully into the future, maybe be able to find markers that show that this progression is happening early enough, so maybe we could slow down the progression of some of these processes and maybe even reverse them. So by that I end, and I thank you very much, and I'll be happy to answer your questions.